Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the bioinformatics. In this particular video, I want to focus on the coding regions of the DNA and how you can use these coding regions of the DNA to pretend to work with the protein sequences. Now, of the hundreds of thousands of the protein sequences that are found in the current databases, only a small percentage correspond to molecules that have been actually isolated by somebody or experimented upon. What I mean by that is that determining the sequence of the protein is much more difficult than the sequencing of the DNA. So finding out the sequence of the DNA comparatively is an easy process as compared to determining the sequence of a protein. As all of the proteins that a given organism, whether we talk about the human beings or the microorganisms, so all of the protein that a given organism can synthesize, they are actually encoded in the sequence of the DNA of its genome. What this means is that the information is stored in the DNA and then that is translated into the protein. So a very smart shortcut that the molecular biologists have been using is to read the protein sequences directly at the information source, that is in its DNA sequence. The codon that are encoded in the DNA, the translation machinery is reading these codon and then you are going to get the primary sequence of a protein. So what the molecular biologists do is that they are going to read these codons and directly find out the protein sequences that these DNA codons can code for. So this way we can pretend to know that amino acid sequence of a protein that have never been isolated in the test tube. So we will be virtually having a sequence of the protein and that particular protein have not been yet isolated in the test tube. They may be isolated in the future to give you the information. Now how you are going to turn these DNA into protein? For that, you have to use the table of the genetic code. We will discuss that in detail in the next video. But what you can actually do is that if you know the sequence of a DNA, you can translate it into the corresponding protein sequences by using the famous genetic code table. The very same way the cell itself generate a protein sequence. So when you talk about the living cells, the cells in vivo, what they do is that they use these genetic code information that we have drawn in the form of a table. So the same sequence the cell uses to make a protein sequence which is corresponding to the DNA sequence that has been there in the genome of those particular cells. Now this genetic code or this genetic code table that is universal, of course there are some exceptions, but most of the time it is universal and it's nature's solution to the problem of how one uniquely relates a 4 nucleotide sequence to a suite of 20 amino acids. So there are only 4 nucleotides in the DNA like the adenine, thymine, guanine and cytosine. 3 of them like this is 1, 2 and 3. 3 of the nucleotides are going to combine with each other to make a codon and one codon is coding for a single amino acid. So how the 4 nucleotide sequence is then coded into a suite of 20 amino acid, for that the cell is using the information that is there in the genetic code table. So understanding how the cell does this, that was one of the most brilliant achievement the biologists have discovered back in 1960. So in the next video, I'll be focusing on the genetic code table, how you can use that particular genetic code table when you know the DNA sequence, so how you can translate that into a corresponding protein sequence. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.